Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Alex Cagan, who is a Postdoctoral Fellow in the Cancer, Aging, and Somatic Mutation program at the Wellcome Sanger Institute. He investigates evolutionary processes in somatic tissue and also does scientific live sketching and illustrating, which we're going to talk about today. And I've seen him do on a number of occasions. If you don't follow him, you should check him out on Twitter. He's got an amazing combo of science and illustration, right brain, left brain going on. As someone myself who's colorblind, I can't draw to save my life. I think this combo is pretty cool. We're going to go into detail on both of these today, both Alex's science as well as the inspiration and an origin story behind his artistic talent. So Alex, thanks so much. And I really appreciate you taking the time today.
Yeah, pleasure. Thanks for having me. I really look forward to talking about the science and the art.
So in an interview in 2019, someone asked you about the most surprising discovery you've ever made. And at the time you said it's not published yet. It comes from a collaboration with the London Zoo. So it involves a lot of cool animals. And I don't know for sure, but I'm pretty sure that you're referring to the work you published not too long ago, a couple of weeks ago in Nature. Am I right about that? And if so, maybe you could tell us about this surprising discovery.
Yeah, wow, good research. Yeah, I was wondering what I'd said by then. Yeah, we must have been pretty excited because we just got those results. Yeah, so we've for the last kind of 5 years, I've been doing this collaboration. It's been years in the making because it's required a number of technical developments to get there. But essentially, the question we wanted to ask was, we knew from work in humans that somatic mutation is a thing, that our cells are accumulating mutations as we age, and that this has a role, we know, in cancer. Kind of these mutations can drive cancer, and also potentially it's been hypothesized they could have a role in aging, because could these mutations be like degrading the function of our cells as we age? And that's been a long-standing hypothesis, but there's not been any good data to test that. And we have human data now that shows our cells are accumulating mutations kind of linearly with age, but that can't really address the question of what role do they have in aging. And we also know, if you look across the animal kingdom, there's a huge variation of species at different ages. So kind of obvious question is, if mutations are playing a role in aging, are species with longer lifespans accumulating those mutations at a slower rate? Is their kind of mutational clock ticking slower? And a kind of second amazing thing, for switching from aging back to cancer, is that some of these large mammalian species, based on everything we know about cancer in humans, like shouldn't be able to exist. Like if you model cancer risk in a whale that has quadrillions more cells than we do, some of these whale species should be getting cancer before they even reach 1 year of age, but they don't. So this is like this classic Peto's paradox, named after the statistician who observed it, that cancer rates kind of stay constant across the animal kingdom, whereas we would expect they would increase. And so one of the kind of resolutions to that paradox is, again, could it be that those larger, longer lived species are better at repairing their DNA, or for whatever reason, they're accumulating mutations at a slower rate? And so kind of we were wondering, are cancer and aging two sides of this same coin, both caused by accumulation of mutation? And a kind of a great way to address that would be to look at these species with different lifespans and cancer risks and just see at what rate are they acquiring mutations. But at the time, there was no real data on this, because it's very technically challenging to measure and actually look at these somatic mutations. It's only recently become possible in humans from healthy tissues. So it's kind of, there's a whole technical development. I can go into that now, I guess.
What has changed technically between then and now that you weren't able to do, but now you have been able to crack this code a little bit?
Yeah, so it's been quite a fun journey. So I was really lucky, just as I was starting my postdoc at the Sanger, kind of starting to develop these techniques. And what it comes down to is essentially developing this pipeline that integrates histology, laser capture microdissection, and low input genome sequencing. So the kind of the real challenge is that traditionally in cancer genetics, you could just sequence tumors, and all of the cells in the tumor will kind of share that initial driver mutation. So you sequence like thousands of cells, and you see the tumor driver mutations present in all of them. So you can easily distinguish it from sequencing errors, which are kind of very rare. But if you look at normal tissues, they're very polyclonal. You don't see the same mutation often across many cells. So to actually distinguish from sequencing error is a challenge. So the way we got around that was, first you kind of take a tissue sample, prepare it histologically on a microscope slide, and then you use laser capture microdissection, which is like the most fun lab technique I've ever done, because you're sitting there with a microscope with a high-powered laser beam on it, and you just kind of select out small groups of like a few hundred cells. And the hope is that then those cells, because they're close together, will be they'll be clonal, they'll share the mutations. And that kind of guides the tissues we look at. So we look for tissues in the body that probably have a clonal structure, like I'll come to later, these intestinal crypts. And by microdissecting them and sequencing them, then we find the real somatic mutations at high enough frequency that we can distinguish them from artifacts. So this was the pipeline that we used. It had been done kind of a year before successfully in human. I did some work looking at kind of building this atlas of somatic mutations in different tissues in the human body. And so then we felt like, okay, we did it in human. Now we can try and move to other species and see if that works there. And then that led on to this challenge of, okay, well, how do we get tissue samples from other species? Because with humans, we can work with like the NHS, and there are biobanks. When you want to look at like just a range of different animal species of different body sizes and lifespans, that becomes more challenging. So then I had to set out on this quest to find people who had animals and get access to those tissues.
What does it take to collaborate with a zoo or with the London Zoo? I assume they've got research teams if they're at a particular scale, but they may not do much genetics, or maybe they do. You tell me, what does that look like? Do you go to them with this crazy idea and they say, it sounds good to us?
I mean, that's pretty much it in a nutshell. Yeah. So obviously, so London Zoo is great. They're part of this Zoological Society of London. So they're like a research focus as well. There's a whole organization. There are researchers kind of affiliated with that. They have these two pathologists there who we work really close with. So their day job is kind of when an animal dies of natural causes, they'll do a kind of necropsy and figure out what was the cause of death. And they were very happy to kind of contribute. You know, it's great if these animals can also be put to use for research that could help with better conservation or understanding these animals better for better care in the future. So they were very helpful. And obviously, like, they're the experts. I wouldn't know if I had to collect these tissue samples, I wouldn't even know where to look in the animal. So they're great, and they collect the samples for us, little kind of small tissue biopsies, send them to the Sanger, and then we start this whole pipeline. But again, a challenge was we wanted species with very different lifespans, and animals, you know, like cetaceans, elephants, they don't die every day. The whole point when we look at them is because they live a long time, and we don't want to harm any animals. We just want to wait for them to die of natural causes. They kind of had to cast a wide net of potential collaborators to see what tissues are available. There's a few challenges really. So one, some of these animals live a long time. And two, because we want to know what's their mutation rate, we kind of get, when you sequence them, you count X number of mutations, but to know the rate, you have to know how old the animal was when it died. And wild animals don't walk around with like a birth tag on them. So zoo animals are great for that. You have them. We really wanted, you know, blue whales or sperm whales in this study because they're the classic kind of, why don't these animals get cancer species? But unfortunately, we couldn't, as of yet, we haven't found samples where we know the age. We're currently investigating that. There are like these kind of esoteric methods for trying to estimate age based on tooth wear.
Right.
People have told me there's a earwax method for whales. So it's like tree rings. They deposit wax, and you get this like whole like a bit like, or like an ice core you can just pull out of the ear canal and try and age it. But then other people tell me that's not so reliable, which I can believe.
You've got World War II era earwax in there that you can somehow date. Wow.
Yeah. I mean, just as an aside, it is amazing like how we know how old some of these species get. Like how do people know that a whale lives 200 years? Right. And like for bowhead whale, they kind of have a maximum age estimate based on a harpoon that was found in a whale. And they knew this particular style of harpoon. It's called like a bomb lance because it had explosives in it. It would go into the whale and then explode. Was only made for like 5 years and like sometime in like the 1800s. And so finding that, they knew this whale had been swimming around for X number of years, and it was probably an adult at the time it was hunted. So yeah, it's kind of mind blowing how old some of these animals can get. So yeah, we've basically tried to get
it together.
Yeah, we pieced it together. We worked with like Cambridge University have a population of naked mole rats who are like they're like superstars or rock star animals for cancer and aging research. For people who don't know about them, they're like
tell us about the naked mole rats. This was one of my questions. Why are they so special and beloved in the cancer and aging community?
They're like beloved in a lot of communities because they're such weird animals. They're like, in any way you look at them, they're kind of different. So they're called a naked mole rat, but apparently they're not a rat or naked truly. Like they have tiny, like really weird name, but they are mostly hairless. They look like a cocktail sausage with teeth is kind of what experts in the field tell me. They're eusocial, so they have a queen, which is really unusual for mammals. There's like a reproductive queen and workers because they live in Africa and they burrow underground. So another kind of rare thing, unusual thing about them. But in cancer and aging, they're fascinating because again, size of a large mouse. Most mice live about 3 years. These animals can live about 27 years. They have an extraordinarily long lifespan for a rodent. And hardly ever get cancer. So for a long time, it was thought they just didn't get cancer at all. But people have been studying so many of them for so long now. You do see there's 2 or 3 reported cases of tumors developing, but it's extremely rare. and so low that they must have some mechanisms of resistance. And again, the reason they're in Cambridge isn't even to do with any of that. It's for a group in pharmacology who studies the fact that they lack certain pain receptors. So I think partly because they live underground in this kind of very CO2 rich, very cramped environment, that they've kind of lost some of to make their life a bit more pleasant. They don't detect all of the discomfort that you would feel if you were living that way.
Oh my gosh.
Yeah. So we got some samples from these guys. So all in all, we had 16 species, including human. So 15 other mammal species with a range of body sizes and lifespans. And naked mole rat was really a key species for us as well, because our aim was to kind of disentangle, okay, we're going to look at mutation rates and we want to see if there is variation, what's explaining that. And like, is it body size or lifespan were kind of two key traits, but most mammals, they get bigger and they live longer. So naked mole rat was quite nice to disentangle that. You've got a species that lives a long time, but it's still very small. So
yeah,
that was our data set.
So the biggest mammal in the study was that the elephant?
We didn't, there was giraffe. So again, even elephant, we'll get onto it later, but now we're in a great space of getting all these new tissue samples. But at the time, the largest we had was giraffe. We still had about 40,000-fold variation in body span from mouse to giraffe. So there's still enormous variation there.
And what were some of the examples of animals in between? So the mouse at the small end, giraffe at the large. What were some of the? I won't ask you to name all 16 unless you can.
Yeah, I could try. The most memorable for me was the Siberian tiger, because that was the first sample we processed. So that was that's a large animal. We had African lion. We have horse, ring-tailed lemur. So it's a primate, which was interesting as well. A whole range. We did have one cetacean, the harbor porpoise, but it's a small one. It's kind of the smallest cetacean. So it's like a small dolphin. And then we had a whole range of like other we had rat as well, ferret, rabbit for the kind of shorter end. We really wanted to get a breadth of lifespans. Yes. It comes out, most mammals, it seems, live between, a few years to 20 years. You get a lot of species in that range. And then it's really a challenge to get the older species.
So with the inclusion of the naked mole rat, you could deconvolute the lifespan and size. So what did you find there? And then after that, I do want to come back to the intestinal crypts and actually how you did the measurement of the mutation rate, because I think it's quite a cool method.
Yeah, sure. So I'll jump to the main finding. So what we found when we looked at mutation rates across these species was, first of all, there was enormous variation in mutation rate. So within like human cells, for example, there's also a range of mutation rates depending what cell you look at. But for the cells we were looking at, the intestinal crypts, in human, you get about just under 50 mutations a year, which is just in itself is extraordinarily low number, I would say, because these cells divide kind of once a week. Every time they divide, they have to copy about 6 billion base pairs of DNA.
Wow.
And what this tells us is for every 6 billion base pairs they copy, they're making kind of just under one error. So it's remarkably low. But because we live so long, in a human, by the end of your lifespan, you have about 3,200 mutations in these cells. Whereas when we looked at mouse, we found they're getting way more mutations a year, like 800. It's a massive number. And you see this variation from kind of 50 mutations per year in human to 800 in mouse. And then what we found that was kind of remarkable is then if you kind of look what's explaining this, it really seems that most of the variance is explained by lifespan. So 82% of the variance. So essentially all of these animals, despite having very different mutation rates and mutational clocks are ticking at different speeds, there's some kind of evolutionary constraint there because all the mammals are ending their lifespan with approximately 3,200 mutations, which is just like, we were kind of blown away by that. We thought it'd be exciting if there was some kind of relationship to body size or lifespan, but it really seems that it's lifespan that explains all the variance. Once you correct for lifespan, you don't see an additional effect of body size. And kind of like a nice way, I think, of like driving this point home is if you look at the naked mole rat again and the giraffe, two species that vary like tens of thousand-fold in body size, but they live almost the exact same lifespan, and their mutation rate is almost exactly the same. It's really the mutation rate that seems to somehow be connected to lifespan.
So I'm sure you've gone through all kinds of potential explanations. The thing that popped to my mind is if the mutation rate in, say, the whale or the giraffe or the elephant, one of these long-lived gigantic species, if it was higher, then to your point earlier, you'd get cancer earlier. You might get it before reproductive age. That's maybe one of the hypotheses. What were you exploring going into this? And do you have a sense of why there seems to be this ceiling of how many mutations an organism can tolerate?
Yeah, I mean, that's really the $1 million question. So we put a lot of thought into what what this could mean for aging and cancer. And if people are interested, we have a whole supplementary note in the paper about it. But kind of what we think is probably unlikely is the classical view of mutation aging is the somatic theory of aging, that just every cell is independently accumulating mutations and they function less well, as because you're just kind of destroying the blueprint of your DNA, which tells your cells how to behave. But this number of like 3,200 mutations, if you look at how many of those are actually encoding regions of the genome, it's only a handful. And we think it's unlikely that just a handful of these mutations could kind of really be explaining aging, which we think is anyway a multifactorial process. But even so, like, could just a handful of mutations really be causing aging in each cell? So we think what our kind of working hypothesis at the moment is that what might be happening is it's not that every single cell is kind of running itself down with these mutations, but maybe it's just a few cells in each tissue do happen to get these mutations that will kind of boost the cell's fitness. That cell will then start to trigger proliferation, kind of no longer doing the role it's meant to do in the cell and start to spread, a bit like the early stages of cancer. So what could be happening as you age is a few cells start to colonize the tissue. And we know this is happening from studies in humans. You do get these clonal expansions of certain cells in our tissue as we age, which are starting to proliferate. And they may be kind of increasing their fitness at the cost of your organ function. So your organs may function less well over time because like these small cells are slowly spreading and taking over. That's our hypothesis. What we lack is kind of, we know these clonal expansions occur, but we don't know, are they really affecting tissue function yet? Because we haven't had the ways to measure that yet. So kind of future aim is to kind of understand, are these processes that are happening in our body as we age, driven by mutation, deteriorating the ability of our cell or organism to function?
How would you test something like that? Do you have a sense?
Yeah, well, I guess there's lots of different ways. What would be really nice is we're getting lots of molecular data, genetic data. What would be really nice is to get some phenotype data on tissue function. So, there are these great kind of organoid models that are being developed, kind of where you can grow skin epithelia in a dish, for example. You could then trigger these clonal expansions and then just try and measure. And I guess the measurements you would take would vary depending on the tissue. But, processes readouts that show that the tissue is functioning normally, are they decreased? Are they negatively affected by these clonal expansions? Is there some connection there between the molecular and the output function? But I think we're still away from getting there, but that's kind of, I think, the direction where we're going.
Fascinating. And I wanted to go back to the intestinal crypts, because I think this is a very it's an elegant way of studying this. I wonder if you could tell a little bit about the process, what it takes to do that, and how you're able to. I guess this is part of the laser microdissection story. So I'll let you talk about that piece of the puzzle.
Yeah, exactly. So we knew for this study, because the mutation rates vary from cell type, we wanted one particular cell type to look at that was comparable across mammals. And we picked these intestinal crypts. For anyone who doesn't know, they're kind of these U-shaped structures that lie in the intestinal epithelium in our gut. And what's great about them is, one, you can see them under the microscope. So you can be like, there's one crypt, I can cut it out. But two, they're just great for calling somatic mutations, because the base of each crypt has a stem cell population. And over time, one of these stem cells kind of drifts to take over the population of stem cells, and they divide every week. So you have these transient cells that are daughters of the stem cell that line each individual crypt. So if you cut them out and sequence them, every single cell in that crypt will share the mutations that have been accumulating in that stem cell. So what you're really getting is kind of a single cell somatic mutation rate estimate, but you get the chance to see it in like a few hundred cells, which lets us then distinguish the true somatic cells from the errors. So they could be kind of just little perfect units you can cut out, and each one will kind of have been accumulating its own different mutations. So the idea was, like in a tiger, you cut out 5 separate crypts, sequence them, and each one will have different mutations, but the number of mutations is the same across all the crypts. You can so it kind of again validates that what you're seeing are true somatic mutations.
Very cool. And by the way, everybody, if you're struggling to visualize this, or if you want to go in more detail, on Alex's Twitter at ATJCagan, ATJCAGAN, there's a really great tweetorial that goes through main figures of the paper. Beautiful. I guess that was that the cover of Nature that you got, this beautiful clock with did you draw that? Did somebody else draw that?
No, I drew that one. That was a lot of fun.
Incredible.
Yeah.
So yeah, there's a 24 tweet thread that goes through with beautiful illustrations, pictures of the figures. You can see all of this in action, and I think it's really worth taking a look, because it's a beautiful piece of work, and will probably help to close the loop if you're struggling to envision crypts, and it's also got a list of the 16 animals on there if you want to go back and see. Is it the saber-toothed tiger? No, saber-toothed tiger. Was that the first one that you sequenced?
Siberian tiger.
Siberian tiger. Saber-toothed tigers are extinct for many
Yeah,
years.
we haven't done ancient DNA. I want to try and combine ancient DNA one day to do kind of look at somatic mutation in saber-tooth.
Yes.
That would be a cool project.
What looking back on the last 5 years, what did you learn from, like, taking the science away from it? This must have involved like loads of different collaborators. The science has changed a lot in the 5 years, new techniques. What has that been like, just pushing a project like this for almost 5 years? It sounds like
it's been a really fun journey. I think I've got really lucky that like somatic evolution, I think, is like exploding as a field, because it just helps us address so many long-standing questions about cancer, about aging and age-related diseases, which have kind of been a bit intransigent to study. We've been very good at, I would say we've been much better at treating infectious disease than we have these kind of age-associated disorders. Obviously, in the kind of biotech space, there's a lot of interest in aging now. And I think these tools have it's just a great time to be doing this kind of biology, because there are these theories that have been around for decades or longer about causes of aging. And now we actually have ways we can generate data to actually quantify and address these different causes. And, somatic mutation is part of it. There's also people working more directly on kind of epigenetics or protein turnover, all these other aspects of aging. And just kind of I think we're now at this stage where we're close to being kind of pieced together and kind of solve this mystery of what are those causes of aging it's like a great biological mystery that's been around so long. And it seems like it's accelerating the technological developments, because when I started, we were just like, can we call somatic mutations in healthy tissue? Right. And now it's more like, okay, which cell do we want to look at? Not just looking at mutations, we're now also looking at these kind of clonal expansions and other kind of selective processes. So I feel like it's like when you've just developed a boat and there's a whole landscape in front of you. Now you just have to decide which way you're going to sail. And there's just so many places we can start to look at. So the problem really is more now picking like what are the key projects to look at, because there's so much unexplored space. And that's certainly a growing challenge with the species, because now we know that we can look at somatic mutation across species. We looked at 16. There's a lot more species out there. Yes. And beyond mammals, if you want to look at aging, there's so many good species one could look at that either, live for hundreds of years, or they're like quote unquote immortal. So there's so much space where we just want to generate that basic data that will help us understand these processes in such a higher resolution.
What are some of the quote unquote immortal
organisms? Well, obviously I've been looking at recently species to find collaborators for, and every time I see a species that's called immortal something, I obviously have to look it up, because if you're interested in aging, you want to look at these.
You've got to at least give it a look.
Exactly. So there's the ones I'm aware of. Maybe people hopefully, if they know others, contact me on Twitter. But there's the immortal hydra, which is like this tiny microscopic organism that lives in fresh water. We're collaborating with a group at UC Berkeley that cultures these. And yeah, they're called immortal. I think it's based on the fact it's a scientist kind of kept them on his like window shelf for like 20 years and never saw one die of old age. And every 20 days, they renew all the cells in their body. So they're constantly turning over cells. And yet they have this amazing ability to just keep on doing that. It's kind of, based on our kind of results from this paper, you would expect an immortal organism like that would never accumulate a somatic mutation. It would have like an insanely high fidelity DNA repair process. But it's hard to imagine this microscopic organism. This kind of like, it's so thin, I think its whole body's exposed to UV light.
Yes.
Not being overwhelmed with mutations. So we really want to sequence that and understand what's going on there. And that will help us understand, we hope, that all the data we're collecting in this kind of new perspective. There's also there's an immortal jellyfish. But I think it's marine organisms where there's a lot of cool stuff going on. And so next week we're getting our delivery of a Greenland shark from the cetacean stranding investigation program in the UK. So this is the longest lived vertebrate species. Estimates that it can live up to 500 years. So we're starting to get into some really interesting species for the aging research.
So in this, are you going to be able to get a sense of the Greenland shark's mutation rate from this data, hopefully?
That's the plan, yeah. And kind of the cool thing that motivates us from the recent research is that human was the oldest species we looked at, but what it would suggest is that these longer lived species should have even better DNA repair, or that's what we hypothesize and we do. And so if we can start to study those species and potentially understand what are the mechanisms by which they're better able to repair their genome and kind of reduce their cancer rate, one day, could you ever take that knowledge and use that for kind of developing therapeutics or ways to ultimately increase our health span? That's kind of that's one of the like end picture goals.
Yeah, so that's one of the things I was going to ask next. And I think you have a slide in your tweet thread about TP53 and elephants. What are some of the ways that organisms figure out? Is it about a lower mutation rate, or is it about DNA error correction, or is it a mix of both?
Yeah, so great question. And I think we're still trying to figure that out. Yeah, so obviously part of our results, they suggest that this reduced mutation rate is probably part of the story for reducing cancer risk. If you have less mutations, it can take longer to knock out those genes that cause tumor formation. But because we don't see this additional effect of body size, it suggests those larger species must have additional mechanisms as well. And as you mentioned, like, yeah, there's some great research on some of these species like elephant, where elephants, it seems, well, in humans, you have two copies of this gene TP53. When you both get knocked out, it's normally bad news. It's normally an initiation step for cancer because it dysregulates apoptosis among a number of other things. And it seems elephants have evolved like 40 copies of this gene. So one hypothesis is that, yeah, I think the number of those genes increases if you look at different elephant species of different body sizes. So there's a nice correlation there. So the hypothesis is it's like having extra batteries, like backups. It's just going to take longer for them all to be knocked out. So if you combine slower mutation rate and more copies of those driver genes, that seems to be, at least in the elephant, one of the ways that they've reduced cancer risk. But you don't see TP53 expansions, as far as I know, in a lot of other large-bodied species. So it kind of suggests, and it may be not surprising, that evolution has found different solutions depending on which trajectory you go by, which is, again, really motivating, that it's worth looking at all of these different large-bodied species, because each one may have a different mechanism for reducing cancer risk, and maybe one of them is going to be much more easy to apply in a human context than others. So it's like an untapped gold mine, potentially, essentially, for discovery.
Yeah, that's fascinating. I want to take another trip down memory lane, because I was doing some background research on this, as you can tell, and I found another article from June 2018 where you went on a trip to Chernobyl, which is a place that most cancer and aging researchers know a lot about but stay pretty far away from. I think you can correct me if I'm wrong on this, but a short trip is probably not a health risk, right? But what did you do there? What was the purpose behind your Chernobyl trip?
Exactly. Well, that's kind of another kind of bizarre tangent of cancer research. It was looking at transmissible cancers, and I'll kind of explain why this ended up going to Chernobyl. So there's a researcher, Liz Murchison at University of Cambridge, who specializes in these transmissible cancers. So in humans, cancers aren't transmissible, really. You have a cancer, but it doesn't spread to someone else because the immune system recognizes it as foreign. But in Tasmanian devils and in domestic dogs, cancers manage to become transmissible. There's these strains of, for dog, it's a sexually transmitted cancer. So it's a tumor that spreads during sexual contact. And for Tasmanian devils, it's on their faces. And there are species that fights a lot, or they kind of snarling and biting, and it transmits that way. And it's still kind of a mystery why in these species it's able to do that. Why doesn't the immune system recognize it? Kind of ongoing field of research. But anyway, for the dogs, this is found in kind of stray dog populations around the world. And they're in the process of just trying to collect samples, because you can map it like a phylogenetic tree, the way it's spread in some kind of weird way. These tumor cells for the dog are like the oldest living dog in the world. It's like a 10,000-year-old dog cell that's become a kind of immortal cancer, and you can kind of look at how it's spread and sequence it and track it that way. And so Liz Murchison was interested in if you go somewhere like Chernobyl, where there maybe is radiation and it's going to be mutating faster, will that kind of have accelerated the evolution of the cancer? Because another weird thing about these transmissible cancers is they tend to have a very low mutation rate compared to most cancers, which are not often become hypermutators. These long-lived transmissible cancers have kind of stabilized and slowed their mutation rate, which might be one of the ways that they can survive for so long without their genome just imploding. So yeah, it's kind of this weird, interesting idea that maybe in Chernobyl, the radiation will have caused faster evolution, and then we could learn some things from that. So so I went out there. I think the person who was going to go couldn't make it anymore, so I thought it sounded like an interesting trip.
That's great. Totally random question that I should know the answer to, but I don't. Do non-mammalian animals get cancer? Where is cancer in the tree of life?
That's a good question.
Where does it start?
Yeah. So I think the kind of claim is that cancer is wherever there's multicellular organisms, you should get cancer, because it's kind of the breakdown of multicellularity. I mean, no one's looked at every single species, but I think invertebrates get it in London. Yeah, like insects get it. I think almost any species you look at, you'll find growths. Well, I guess it's hard to be sure those are cancers without sequencing them. Like often in trees, you get these growths, but we think they're not actually cancers. They're induced by a parasite, to stimulate growth. So theoretically, I think any multicellular organism should be at risk of cancer, because it's almost like you can think of it as almost a bit like a reversion to unicellularity. It's a cell that starts to break. Every cell has these rules how to cooperate in the body, and when they start to break down, and the cell becomes, quote unquote, selfish. There's a new book by Kat Arney called The Rebel Cell, which is kind of a popular science book all about cancer evolution. And you can kind of think of it that way. It's these cells that go rogue. And so why shouldn't that happen in any multicellular organism? If you get the right mutations that break down those genes that are regulating cooperation, it's kind of the risk that you have of being a multicellular organism.
Yeah, fascinating. And if anyone is interested in Rebel Cell, Kat came onto this podcast a long time ago, episode 46, right when the book came out. So you can hear straight from Kat on a couple of weeks after release. And I read she sent me a copy of it prior to the episode, and I highly recommend it to everybody. It's a really fascinating read.
Yeah, absolutely.
When did you first discover your talent for? I want to hear about when you first discovered your talent for illustration, and then also when you first applied it. Which came first, the illustration or the science? And when did the two worlds collide?
Yeah, it's a good question. I think generally the illustration came first. So I was at school, I loved biology, but then ironically, when I was like 15, I got put off by genetics because I loved animal behavior, and then suddenly I went from animal behavior and like, oh, I loved all of this, like looking at images of like cheetahs on the savannah, and then it switched to Punnett squares and like equations, and I was like, this isn't quite what I was into. So I ended up kind of focusing more on the humanities, and my mum is like a set designer, illustrator type person, so I've always been around that and more encouraged with that. And yeah, so I went down a humanities route, studied anthropology at university, and I wanted to get into kind of social anthropology and understanding. I was still interested in diversity and differences, but I was looking from the kind of more human behavioral perspective. But during that course, you had to take modules in genetics, and you like to look at biological anthropology. And then I kind of rediscovered my passion for that. And again, it was at the time when next generation sequencing was just emerging. So again, it was very exciting that suddenly we were learning about, Neanderthals and human evolution. And so kind of got back into it that way, but kept going with sketching. But they were two separate paths. I never thought about linking the illustration with the science. So that came about during my PhD. I went to a symposia, SMBE meet, like a Systems for Molecular Biology and Evolution meeting. And again, Twitter was just taking off. And I had an iPad mini, and I saw these people like you and Bernie type, like tweeting away, really good like coverage of the meeting. I was like, oh, how could I do that? But I thought, no, there's already these people doing these great Twitter threads. And I realized they were always like, you have a thread because you can do 140 characters per tweet at the time.
Yeah.
But you can completely cheat by sharing an image.
Right.
I was taking my notes, just like doodling, and it would probably take hours. If you scroll all the way back to the beginning of my Twitter, the first images must be there, and they were like terrible. It was just like I'd write like maybe the name of the talk
and draw a little something,
and it's a little something, like just like a little thing of their face, or a little picture of the animal they were working on. But then people liked them enough that I was like, oh, this likes, I'll keep doing this. And it kind of evolved that way. And I think also Twitter is kind of a useful, has like an evolutionary mechanism, because you can kind of see how many likes things get. You kind of put, okay, this is the kind of content people enjoy. And it kind of just evolved that way. And I also have a terrible attention span. And sketching really keeps me focused. It helps me think, it helps my mind from drifting. So I found it really invaluable. I was actually recording and understanding more from the conferences than I would have if I wasn't sketching. And then just sharing them is so easy, right? You just click share on an iPad and it's out there. So I thought, why not share it as well?
Yeah, and we'll it was like, it's like the ultimate badge of honor when you get drawn by Alex Cagan at a conference. It still is this way, but I remember there was a period of time in like 2018, 2019 where people were just hoping that their talk would get picked up and a beautiful illustration was drawn. So I think it's an amazing thing. The point about Twitter is a really good one, because it for a long time it wasn't a great place for visual learners. If you wanted to read a tweet thread, that was great, but a picture is worth 1,000 words sometimes.
No, absolutely. I think it's more recently, like the evolution of tutorials has been great too. Like again, so few people, as people are so busy, like often don't have the time to read the paper. You get a lot more with, I think with a good tutorial, you get so much more exposure. And even people who will read the paper, having like a clear, it's like a long visual graphical abstract. It just helps people to immediately get the take-home messages and to share it quickly with other people in a much faster way than if you just shared the paper. That takes a long time for people to, how many people ever read beyond the abstract anyway? So actually to have that format, I find really useful for people to engage with, and as long as they're getting the key messages that way, it's great if everyone can read the paper, but that's just unrealistic. So at least if you can deliver the message there in a clearer way than just in an abstract, I think it's a really useful medium for sharing new results.
Have you been playing around with Midjourney or Dolly or any of these fun new tools?
Yeah. I'm waiting until I can just give one all of my illustrations and then just get it to immediately. I'm sure it's not far away when they'll be out live feeding me. It's great. Yeah, it's amazing what they're doing. I'm kind of, I guess a lot of illustrators are nervous. I'm kind of excited to see where it goes. Like there's just so much potential there, and I think anything. There's two sides to it, right? You want to make sure that creators and illustrators still have employment and things they can do, but also the potential now for people to just have these tools for people who aren't into illustration, who can't afford an illustrator, to help visualize research. I'm just really big on science visualization and using that as a tool for communication. I think there's just so much potential we can't even imagine now for how that's going to be able to help people to communicate scientific ideas and make them more compelling.
Yeah, I think that's a really good point. If we separate there's clear copyright challenges, and we need to figure out how to not have the tools like completely rip off somebody's style without the right credit. But on the flip side, I think it democratizes access to the tools in a really interesting way, where if you can describe what you want to be drawn, you don't need to be able to do it yourself. Hopefully the machine can get you 90% of the way there. But I think there's always going to be space for people who have clear artistic talents to tweak and make breakthroughs, right? I think these tools are good at synthesizing, but not so good at discovery yet.
Yeah, exactly. Yeah, that's what I hope, that it'll just be a powerful tool that will just enable a lot more great science visualization. Ideally. Yeah.
I think you're really onto something with that, and I love it when great scientists like you are also equally committed to creating digestible formats for people to understand what you're doing, because there's too much good science that only lives in the small corner of the universe that has the time and background to read the paper. So it's really great when you can spend the time to distill it out for everybody.
I just wish more people. And I think more people are doing it, but I just hope it becomes more popular, because it's again, like for areas I'm not an expert in, having things like visual tutorials or any kind of visualizations. I'm definitely a visual thinker or learner, so it just helps me so much. And you can condense so much information in a good tutorial so well.
Yes.
Big fan.
Yeah, I tell everybody to go follow Dr. Vera as well. If we can combine his tutorial skills with your illustrations, you guys will be unstoppable.
That'd be awesome.
I just as a final closing question here, I wanted to pick your brain about the Tree of Life study and studies. I think there are a couple going on around the world, but for those who aren't familiar, there's at least the one I know of at the Sanger, there's an incredible study going on to sequence, I think it's more than 100 species across the Tree of Life to I think get at some of the questions you were pointing out earlier, not just in cancer and aging, but at a very fundamental level. What do all the animals and organisms in the world around us, what does their genetic blueprint look like, and what can we learn about that, both from fundamental science but also applications to health? Or I imagine you must be excited about that, but what are you excited about? What is that going to enable and allow us to do in the next couple of years?
Yes, I'm really excited about that, and that's one of the great things about being at the Sanger, that there's the Tree of Life program, and within that the Darwin Tree of Life project, which aims to sequence every eukaryote species in the UK, and there's initiatives around the world doing similar things. So yeah, in the next couple of years or decades, should be a reference genome for every multicellular organism on the planet, which is kind of mind blowing. And yeah, what that does for us is, I mentioned this intestinal crypts and how we had to look for these clonal units. So now we have a new method, duplex sequencing based, called NanoSeq, that's so accurate we can sequence, it doesn't have to be a clonal unit anymore, we can sequence at single molecule level. So we have the ability, now we can look at any cell type, and combining that with the Tree of Life programs, making reference genomes, we're in this position for the first time where we can look at any cell type in any species on the planet And so, yeah, just again, it opens up this whole unexplored landscape where we're not limited by, we used to be limited, like you have to look at human laboratory mouse or C. elegans, Drosophila. Now we're not limited by those, and those species are great, but for so many biological questions, they're not the, if you were picking a model organism, you wouldn't say, okay, I want mouse. There's so many other species there that might be better. And now we can access them and we can look at them, and we can use all the tools of human somatic mutation studies and apply them to pretty soon any species. So that's just I think it's gonna lead to so many new cool discoveries.
Amazing. Well, Alex, thank you. I really appreciate all of the amazing work that you've done on this paper, taking the time to explain it to us. And I would really encourage everybody go follow Alex on Twitter. A lot of good stuff. Thank you so much. I really appreciate you taking the time.
No, my pleasure. It's been great to chat about.
And thanks everybody for listening. As always, we really appreciate if you could share the podcast with a friend or leave us a review on your favorite podcast player. Thanks again, and we'll see you next time.